So I'll be giving my talk on substrate loss modulus promotes the differentiation of neuroblastoma cells. This work was carried on in IIT Bombay as a part of my PhD program. So I'll be discussing this work. Before going into the details of the nitty-gritties of science and the experiments involved, I'm going to give you a brief idea about what is neuroblastoma. Neuroblastoma is a type of cancer that happens in the neuronal cells. And in the neuronal cells, specifically in the glands, the adrenal glands, the neuron cells of the adrenal glands, or it can occur in many different locations like chest, stomach. It specifically happens to the neuronal cells. Now, as you can see in the neuronal cells, the basic structure of it, it has a soma, it has an axon, it has a dendron. This particular type of cancer happens in the immature neurons. So it happens in the kids, or I would say infants, below the age of 5 years. And it is very prevalent, as you can see, among all the childhood cancers that happen, 6% cause is for the neuroblastoma. So it has its specific position in numbers among the infants. Now, for this, there are specific several treatments for neuroblastoma. So to treat them, you have chemotherapy, radiotherapy, and similarly, other therapeutic modules for that, like some sort of radiation therapy as well. However, as it happens to the infants or the kids, it becomes very difficult for the treatment, as well as this is happening in the immature neurons. So as we grow into adults, what happens? These immature neurons differentiate. There is a process called as differentiation where their characteristics get changed and they start to produce these interconnecting connections. Like every neuron is connected to thousands of neurons to form neurotic pathways. Right. So then immature neuron has not yet made connections. And this particular disease happens to immature neurons. Once you differentiate that neuron, so once the neurons get differentiated, what happens? It has been seen it is a medicine for or a therapy for neuroblastoma because the differentiated cells no longer have the process of rapid division, which is seen in neuroblastoma. I mean, as in cancer, we know what happens. Uncontrolled growth, uncontrolled division, right? So what are the agents that promote neuroblastoma divisions or neuroblastoma differentiation? So if you see these images here, these were images, phase images taken for the neuronal cells cultured. The SHSY-5Y is a neuroblastoma cell. It is cultured in the lab with a certain media, which we call it as the DMEM media. And when you see take the phase images, the phase contrast images of these cells, these are the undifferentiated ones. So if you see the undifferentiated cells, the length of the neuroids are very small. If I can zoom it in and show you a bit, it could get blurred in between because I'm zooming the screen in between. So if you see here, the length of the neuroids are pretty small. But as the cells differentiate, what happens? The neuroids get longer and they start connected to other neurons. And that is what we mean by differentiation. And this is a normal process that happens in the brain, but it happens over a period of time. As the neuron cells grow, they differentiate, they start connecting. And this neuroblastoma is happening in the undifferentiated cells, which have not yet made connection. However, if we could give certain agents or environments where the cells can differentiate, or the immature neurons promote the differentiation, then their division gets inhibited. So in cancer, what is happening? The neuroblastoma cells are dividing more and more because they are undifferentiated. Now, when we differentiate them, what's happening? Their division or the uncontrolled division is kind of stopped. So now we will see what are the known factors that contributes to this neuroblastoma division. So there are several mechanical stimulus, chemical stimulus. Now biochemical agents like when we are culturing the cells in the media as in their growth medium, if you add certain agents like retinoic acid, brain-derived growth factors, neurotropins, phospholesters, they have been reported that they cause this sort of differentiation. And within 3 to 5 days, you will see this sort of differentiation. So it's basically a differentiating, enhancing agents. Now, interestingly, recent research have focused more on not just the chemical agents, because chemically, yes, what happens? Chemicals go and promote the cells into differentiation mode. However, there are mechanical agents. Now, when you put a cell on a substrate, like if you can put a cell, when you're putting on a cell on a glass substrate, it senses the stiffness of the substrate. So when you sense that substrate, the stiffness gives some sort of stimulus. Even if we have seen that when you put cancer cells on a stiff substrate and you put cancer cells on a soft substrate as in, I'll just give you an example here. When you are taking the cells, identify the original stiffness, okay? Like bone, when you touch a bone, you feel it hard because it has a certain amount of stiffness. And these stiffness could be measured through atomic force microscopy. So there is an indentation method where you pinch in the substrate and then whatever force it gets, the returning force, it tells us the stiffness. For example, if you have a mucus, the brain, I mean, you can hold your bone around the bony regions or the elbow cones and touch your cheeks. You will of course identify that cheeks are more softer because it has only the muscle mass, right? Muscle or the skin mass there. So the brain is even softer. The brain has a stiffness of around 0.5 or 1.5 kilopascal. You can see here. So even if we don't go into the mechanical details of elastic modulus and loss modulus, so if I explain you this, stiffness, I think you have understood by just sensing the substrate. So if you compare bone versus skin, and skin is much softer as muscle is much softer compared to bone. The stiffness of the bone of the brain is around 1.5. Now considering viscosity. Now, if you compare water and honey, we know that honey is more viscous, right? Similarly, our brain also has a certain amount of viscous nature. So it has a certain elastic nature, which we measure it through its elastic modulus, and it has certain amount of viscous nature. And everything what I'm saying here, the values quoted here, have been reported using mice models, that what is the general viscosity or the stiffness of the brain. So brain has a certain amount of stiffness nature and the viscous nature, which probably promotes its neurons or the growth process of the neurons. Now, literature suggests, we were talking about differentiation. So literature suggests that biochemical agents promotes differentiation, but along with that, the substrate stiffness, or that the stiffness that we are talking about. So when we put the cells on gels of different stiffness, so for that we will prepare gels, and I'm going to the methodology a bit later. But suppose the cells are put on gels of different stiffness, they're going to sense that stiffness. That much height of the gel has been kept, that they sense the stiffness and accordingly behave. And it has been shown that a bit lower stiffness, like if you compare with glass, and if you compare 0.5, 0.5 is a very low stiffness. And comparatively, as we go to the increase in glass, there is somewhere an intermediate stiffness, which is pretty much like the stiffness of the brain, where the cells get maximally differentiated. That means in that particular micro environment, which is pretty much similar to like brain, what happens? The stiffness of the, or the differentiation of the nerve cells is promoted. Now, similarly, it has been seen that if you look at a cancer tumor or a normal cell, the stiffness of the cancer tumor is a bit more because of the lump and the stuffs formed. The stiffness of that tumor is more compared to the normal. So if you can reduce the stiffness externally, it also affects the proliferation of the cells. It has been seen that if you put cells, cancerous cells, as I said, on a very low stiffness, they will stop dividing. Now let us look at the methodology of the experiment. How did we created a particular stiffness ka gel? So first of all, what is required is you prepare a gel stiffness, and once you prepare it, there is an instrument called as rheometer by which you can measure that kind of stiffness. Okay, so we will come into two concepts here, hydrophobic and hydrophilic. Okay, so how can we make a surface hydrophobic? There are certain hydrophobic agents, and even nail polish also acts like a hydrophobic agent, or things which will not allow water to adhere to it. So we make a glass slide on which is coated on with a hydrophobic solution. Okay, to that certain amount of gel solution. Now, what is this gel solution? It will contain acrylamide, polyacrylamide, the one which we use for SDS, right? Certain amount of that, a certain ratio of acrylamide, polyacrylamide, bisacrylamide with APS, a certain concentration, a certain ratio has to be taken so as to get this particular substrate. So if you see here, I have written the composition here, that if I need 30 Pascal low substrate or 300 Pascal high loss substrate, a particular kind of substrate to identify the viscosity, I will need a certain composition. Now, when we put the gel solution on that, you put a hydrophilic cover slip over that. So hydrophilic means what? The water is going to get adhere to that, right? So once you put that, what happens, and allow it to solidify, it will take roughly around, depending upon your APS concentration and the surrounding temperature, it will take around 20 minutes to solidify. Once it gets solidifies, you can peel it off. Now you will peel it off using the hydrophilic cover slip. Once you peel it off, you will see the gel gets adhered to the hydrophilic one, because the hydrophobic will never get attached to the jelly stuff because it is hydrophobic. It does not get attached to water. So what happens? The hydrophilic cover slips has the gel. Now, on that gel, what we do, we coat the gel with the surface of a protein, because cells won't adhere on that jelly surface until there is a layer of extracellular matrix. So we coat the gel using UV cross-linkers through collagen or fibrin or fibronectin, some sort, gelatin, any sort of extracellular matrix. You have to coat the gel with that, and then you put the cell suspension on that. So in this way, you can have a glass control, and you can have a, in our work, we took low loss modulus and high loss modulus. So it was taken such that the high loss modulus was like the brain loss modulus. So glass is 1, the high loss modulus was the modulus, or as I said, the viscous and the elastic properties similar to the brain one, and the low loss modulus, which I'm representing as LLMS here, has a modulus which is pretty lower than the brain one, right? And glass is not viscous, so it will just have a stiffness. So it was just taken as a control. Any jelly substance only can have that viscous property. So the ideal control is low loss and high loss. Glass is just a general control of differentiation. So when we did that, after that, what was done, the neuronal cells were kept on the, were seeded onto the gels, and there were control cells. We injected differentiation media after 2 days after the cells got adhere, and we allowed to see their differentiation till 3 days. Within 3 days, the cells started making the neuroids, and then the length of the longest neuroid was measured. So if you see this cell, how we took the measurements was like, you have the cell, and then you monitored their length of the longest neuroid. So whichever a cell can have multiple neuroids, we just monitored one neuroid in the cell, which was the longest one. Now, if I show you the images, you can see on glass, this is the control ones, okay? And you can see here, the cell started making neuroids on HLMS even after day 1. Now, what does that mean? You have put the cells on glass, put the cells on low loss, put the cells on high loss. If you put on high loss, you see automatically, just by sensing the substrate, the cells started differentiating, and you have not put any induction media. Induction media as in the biochemical agents. So even without providing the biochemical agents, we saw differentiation on day 1. On day 4, I mean, you can see growth medium, normal growth medium in day 4, and DM is differentiation media, where you had the growth media along with differentiation media. So what happens? You had day 1, where you had just the cells who have sensed the substrate, sensed the modulus, and after that you combine the effect with that loss modulus to the stiffness, I mean to the differentiation media, and you had a combined effect on day 4. And when we had all these, we monitored their differentiation. So if you see here, on day 1, the differentiation has already been doubled, and we can see the neuroids of 200 micron. All these images were taken using phase contrast microscopy. On day 4, in growth medium, there was the low, if you see, the low loss modulus ones. When you give differentiation media, we see an enhancement in that, right? But the HLMS one, we didn't saw an enhancement after 3 days. That means whatever they had to sense in the very first day, they sensed it and they made the neuroid length. But when they were allowed to sense that environment for 3 days, there was no change after that time. But when it was combined with the growth medium, we can see an enhanced differentiation. If you compare it with the very first, which had only 100 micron, and only HLMS, which was around 200 micron, the combined had around 350 to 400 micron long neuroids. Now, this was a sort of morphological analysis, because whatever you see in the phase images, this was the morphological analysis. Now we look for the biochemical impact. This study was done through immunofluorescence, and acetylated tubulin is a marker for differentiation. So it's like the more and more differentiated, you will see more acetylated tubulin being present. So these were the control day one. In day one itself, we see the HLMS having some amount of acetylated tubulin cells getting differentiated. On growth medium, we can see the biochemical differentiation and enhanced. If you compare with the morphological data, there is no enhanced in the length of the neuroids, but the inherent expression, the inherent differentiation of the biochemical markers got enhanced. And the maximum we can see with in combination with growth medium, along with the retinoic acid or the biochemical agents, along with the stiffness. So this combination gives us the maximum differentiation. So all the study was performed on the SHSY cells, and you see, we further see all the study we have done on acetylated tubulin. Now, tubulin is basically a particular subset of microtubules which is more stabilized. So we see when the cells are getting differentiated, microtubule stabilization is taking place. So this stabilization route of the neuroids, so as the cells are forming those strong axonal neuroids, microtubule acetylation. So whether this microtubule acetylation is serving as a pathway to make the long connections, what are the signaling cascade for that, all are the future work for this study. So if there is anything I want you to take from this entire presentation, is two key messages I would like to forward. One is that neuronal differentiation or differentiation of the neuronal cells can help or prevent relapse of the neuroblastoma. It's like a therapy. So if somebody has neuroblastoma, along with radiation therapy, chemotherapy, neuronal differentiation is also a therapy. That is one thing. And second thing, when neuronal differentiation takes place, microtubules are stabilized on a high loss substrates, which is like a substrate having an environment, viscosity, and stiffness, pretty much brain-like stiffness, and it aids in neuronal differentiation. So it is not just the chemical parameters, but also the mechanical parameters helps in neuronal differentiation. So that is something which I would like to take you from this entire presentation. So thank you all. You can ask your questions, and if there are any questions in the chat box, I would like to take them.
Thank you so much, Ms. Nikita, for that insightful session. Does anyone have any questions that they would like to ask Ms. Nikita? Oh, okay. So I have a question. So how did you, what caused you to do your research in this field?
Okay. I can answer this question. Actually, my work is related to mostly drug and cell biology, but my co-supervisor works in this area of substrate stiffness and mechanobiology, Professor Abhijit Majumdar from Chemical Engineering Department. So he used to work on that. So then we collaborated and thought of this project because neuronal differentiation is something which I have studied in my work. But whether substrate modulus or substrate mechanical environment or microenvironment can play a role in that and promote the differentiation, that was not at all known. So this is the first of its kind work where substrate viscosity is playing a role in neuronal differentiation.
So you've never seen this before in any other case?
See, the point is neuronal differentiation has been shown through biochemical agents, right?
Okay.
So chemical agents are promoting the neuronal differentiation. Okay, but the mechanical environment has been known to see like cell proliferation, cell growth, but the effect of this particular differentiation on substrate, it was not shown earlier. So we were quite interested whether it has some effect or not, because recently a lot of papers were working on viscoelasticity. So there was a term called as viscotaxis, where the movement of cells based on a certain stiffness, because cells sense in our body, cells are not static. They sense, they move, they sense their environment. They don't not only sense the chemical agents, they sense the mechanical agents. That's what this entire paper wants to convey.
Okay, it's very interesting. Thank you so much for your presentation. It was very insightful.